Thank you everyone for coming to December's Genomics for Teachers series. This is run by the Nanobio Interface Center. And I'm very happy to have Dr. Dan Simola here today. He graduated from Penn with his PhD in the Genomics and Computational Biology program in 2008. And he's been in the current lab that he's in with Shelley Berger, who runs the Penn Epigenetics program since 2010. And he's an expert on, well, a lot of different things, but in particular, most recently on ants and epigenetics. So without further ado, I'm just going to turn it over to Dan.
Thanks, Kristen, and thanks for inviting me to share some of this with you today. I first want to point out the handouts. I'm not primarily a teacher, and so I'm trying to hopefully hit the sweet spot with not too much detail, but sort of relevant facts that you can take home with you and digest over time. So one is just sort of the slides, and throughout the slides I have a bunch of web resource links that vary from Wikipedia to primary literature. Hopefully everything is freely accessible. And then the other handout is a publication that came out earlier this year, which is a review paper on the use of social insects as sort of emerging biomedical model organisms to study a variety of processes, not just sort of basic research in what are the mechanisms of epigenetics and what are the phenomena, but going out to sort of neurodegenerative diseases and developmental diseases, learning, memory, neuroscience, sort of, and lifespan extension, which I'll touch upon at the end. So, as Kristen mentioned, I'm doing a postdoc in a sort of an epigenetic setting right now. My background is actually more in computer science and genome sequencing. So when I sort of started working with ants, I wouldn't consider myself an entomologist. So when I started working with ants, I brought sort of the tools of genetics and quantitative biology to try and understand how behavior might be regulated epigenetically. And what's I want to say? Well, one thing I want to say is, since you're the teachers, not me, I would certainly appreciate any feedback on things that you found particularly sort of exciting or maybe too detailed. But also, I really got interested in science in general and biology as a high school student. So I sort of appreciate the interest in continuing to bring new ideas to students and get them excited. So, all right, so both of these, this is one of the species I work with. It's a carpenter ant, very similar to the giant black ants that you'll find around the area here, especially on oak trees. These in particular live in Florida, and you can see this dramatic difference in body size is, we believe, epigenetically regulated. And this is a famous example in mice where differences in folate diet, so folic acid, which is what expecting mothers are highly recommended to take by their doctors, differences in folate in the diet of these mice that are all sisters of each other can result not only in differences in coat color, but differences in coat color is just sort of a marker for future diabetes, cholesterol level, and expected lifespan. And in fact, these differences can be induced by high levels of BPA, bisphenol A, which is a chemical that has been used to manufacture a variety of plastics and is sort of coming under increased scrutiny recently. All right, so we're going to spend a little bit of time just talking about sort of the rationale behind epigenetics, the principles, and then sort of what are some of the actual molecular mechanisms behind it, because I think that is, if you get that right, then you can easily sort of communicate the variety of interesting phenomena in nature. Since this is sort of a genomics-oriented workshop, just talk a little bit about the relationship between epigenetics and genomics, talk a little bit about the role of epigenetics in human diseases, and then talk about what's nearest and dearest to me, which is the ants, as models to look at not only epigenetics of developmental differences shown in the previous figure, but also how epigenetics might regulate behavior. So when people ask me what's the deal with epigenetics, the best example I can give them is our own body, right? So the human body, in fact, all multicellular organisms are composed of multiple different cell types, right? We have bone cells and neurons in our brain, and blood cells, and heart cells, and muscle cells, and then we have our gametes, eggs and sperm. So there's over 200 different distinct cell types in our body. All these cells are derived initially from a single cell, which is the zygote, and they all contain exactly the same DNA sequence, right? So how do these genetically identical cells turn into all these specialized cell types, and not only turn into them, but maintain these distinct identities over long periods of time, right? So there must be some additional layer of information above the DNA sequence itself that helps each cell sort of maintain what its identity is. That's called epigenetics, prefix epi just being something above the genome itself. And the modern definition is a heritable maintenance of phenotypic information via non-genetic mechanisms. And I put heritable in parentheses here because there are a variety of mechanisms that facilitate sort of molecular memory that don't involve cell division events, so mitosis or meiosis. So technically, something epigenetic is something that is like a piece of DNA that is heritable across generations. But of all the sort of truly epigenetic mechanisms, they're all a subset of mechanisms that can propagate information just within a single cell for long periods of time. And the best example of that is our neurons. So each neuron in our brain, they all divide initially in early embryonic development, but then there's no more neuronal cell division for the rest of our lives. So they maintain their identity for 100 years. And not only that, they, as presumably the seat of our memories, they remember specific environmental inputs for long periods of time. So the information that epigenetics specifies has to do with phenotype, and phenotype is just broadly defined, right? It could be something like hair color, or the cell type that we're talking about, or down to the molecular level within a cell, which genes are expressed, or what particular behaviors are expressed. So I think the two key questions of epigenetics are, one is that genetics provides the information for how to make a phenotype. It says, here's my complement of genes that can be transcribed into RNA, that can be translated into protein. What kind of information is needed that epigenetics accounts for? And then two, we know that DNA is sort of the molecular stuff of genetics. It provides for that information. What is the molecular stuff of epigenetics? So just to sort of review on the central dogma, we have our DNA, which is a sequence, and specific subsequences of DNA encode genes, something that we call a gene, and that gene can be transcribed into an RNA. And there are a variety of classes of RNA. There are small RNAs, long RNAs, what are called T or transfer RNAs, microRNAs, which actually bind to mRNAs to degrade them. So it's a whole mess of different kinds of RNA. And it's really only one subset of RNA, which is this mRNA, that gets translated into proteins. So our body has 20,000 or so genes that code for proteins, but there are at least that many more genes that don't code for proteins that are also functional, but they function just as RNA. So this is the central dogma of molecular biology, and just focusing on this step of transcription, here is our DNA, and this green bar represents a gene that we want to turn into an RNA, and the idea is that there are pieces of DNA upstream of the location of that gene that other proteins called transcription factors recognize and bind to, and these transcription factors are notable because they recruit the machinery which is a RNA polymerase that binds in front of the gene and then makes the copy into RNA. And what's notable is that recent estimates are that in the human genome, over 90% of it is transcribed, and yet only 5% is encoding for proteins. So there's just this vast repertoire of genes out there, and the majority of them are non-coding. They don't code for a protein. So, and just to review, genetics and genomics is really the study of what these genes are, how do we characterize them, how they're organized in the genome, and how they vary and are inherited among individuals, populations, and species. Now, going back to the idea of the multiple cell types in our body, every cell type is really defined by the genes that are expressed in that cell, and in particular, how many copies of each RNA there are, and then how many copies of each protein there are. And it's really these RNAs and proteins that are the building blocks of that cell, and they also help reinforce the identity of that cell. So you might think of sort of a stem cell that eventually differentiates into all these terminal cell types as, in our, instead of 20,000 genes, I have just 3 here, but it might have a few copies of gene A, a lot of copies of gene B, and somewhat less copies of gene C. But then each other cell type is going to have sort of a different distribution of gene expression levels. And this is sort of one of the bases of thinking about quantitative cell biology. How do you define cell types in terms of the number and abundance of all of the genes that are encoded in the genome? So this, just appreciating this merits the question, well, so then how does a cell know when to turn a particular gene on and off? Because not every gene is on all the time in the cell. So this, back in the first half of the 20th century, led to speculation that epigenetics specifies information for regulating the expression of genes in the right time and place. So this is the original definition of epigenetics, which was coined by scientist Conrad Waddington, who's a professor in Edinburgh, Scotland. And his definition is the interaction of genes with their environment to help bring the phenotype into being, right? So in a cartoon framework, he didn't know what was going on in a cell. In fact, in 1942, the structure of DNA hadn't even been discovered yet. But because of Mendel's laws and inheritance, people knew that there were these things that they called genes that were inherited from generation to generation, and that, especially in Waddington's work, he could show that by treating flies with different environmental exposures, he could get different phenotypes to arise. He was actually looking at sort of the pattern of wing veins in fruit flies. And he showed that certain ether in particular given to juvenile flies would initially induce sort of mutations in these wing patterns. But then if he kept breeding these flies over time, eventually he didn't even need to introduce the environmental trigger, and the flies would just develop abnormal wing patterns on their own without the environmental stimulus.
So this goes back to the nature-nurture argument kind of
Exactly.
thing. And
So,
then what you're saying, then it's almost like then it's not the genes learn, but then they're selected to then
right. So actually
then
his...
not have to be exposed to that environment. They're the ones that propagate it.
Yeah, so that, I mean, sort of a advanced topic, but what he proposed a theory that's sort of yet to be fully accepted or rejected, where so evolution happens on a population scale. And so among a bunch of individuals, each individual is going to have a slightly different set of alleles for all of their genes, right? And so what he proposed was that by imposing a different environment on a population, you're changing the selection of which alleles should be represented the most in that population. And so it's sort of, you have a pre-existing pool of red and green marbles, and let's say 90% are red and 10% are green. By changing the environment and then waiting for enough generations, he saw the increase from that 10% of green marbles up to 60%, right? So the idea is that whatever's happening in the cell or the organism, if you impose different environmental regimes on the same genotype, the same genetic background, you can get these different phenotypes. And indeed, epigenetic processes mediate not only a cell's long-term phenotype, but an organism's long-term phenotype. And these are just some notable examples. One is that this is a small mustard plant called Arabidopsis, which is a model for plant development, plant genetics. And it turns out that the timing of when this plant should flower in the spring is actually encoded the fall before, based on the decreasing average temperature and periods of daylight. So there's an environment, sort of a physical environmental input that changes the structure of, we'll talk more about it, but it essentially turns a gene off and it reduces the number of copies in that cell so that when in the spring temperature starts rising, the number of genes that are left over the winter determine how long it takes before that plant flowers. In amphibians, particular frogs, salamanders, and a few reptile species, the temperature that the brood, the eggs, are reared at determines the sex. So even if, in amphibians, sex is similar to ours in that there's a Y chromosome to make males, but even if you're XY, if you are, as an egg, you're exposed to a high enough temperature for long enough, that temperature will override the fact that you have a Y chromosome and you'll develop into a female. And it's sort of all variable. Sometimes it requires an increase in temperature, sometimes a decrease in temperature, sometimes females will become males, sometimes males will become females. In the social insects, in particular ants and the honeybee, the cast, or the final sort of form of your body that you take as an adult, is determined before. So insects go through, a lot of insects go through metamorphosis, where there's a juvenile phase of development where you're basically a bag of goo, and then there's a trigger that has you go through metamorphosis. So you think of the caterpillar to the butterfly. In ants, you're a bag of goo that doesn't move at all, and then you transform into something like this. And this is called a cast, and depending on the nutritional input you get when you're a bag of goo, it'll turn you into a small miner or a big soldier, or in the case of the honeybee, a worker or a queen. And it's not really known how this happens in ants, but it's very well characterized in honeybee that the workers actually synthesize a special food source called royal jelly that they put into the honeycomb cells that they want to, for the larvae that they want to turn into queens. And there's ongoing analysis of what the particular compounds are in royal jelly, but a lot of them act directly on the epigenetics machinery in a developing larvae. And then finally, I mentioned this example of this coat color is regulated by a pigment gene called agouti. So these are agouti mice, Agouti. All right, so we've sort of answered the first question, which is epigenetics specifies information for regulating gene expression. And now let's take a step back and think about what is the molecular stuff of epigenetics. So not only is the genome, it's not just existing free floating in the nucleus of our cells, but it's actually organized into chromosomes, right? So you have your double helix of DNA itself, and the fundamental building block of the chromosome is what's called a nucleosome. And the nucleosome is a set of 8 proteins that essentially form a ball, like a ball bearing. And then DNA is wrapped twice around each ball bearing. And the DNA and these 8 proteins that form what's called a histone is called a nucleosome. And this organization of DNA wrapped around nucleosomes is sort of canonically called the beads on a string model. And the identity of so there's 2 copies of 4 different proteins that go into this histone, that go into a nucleosome. Each of them is a different histone protein, and each of these proteins can actually receive a variety of covalent chemical modifications that alters the sort of so DNA is an acid, deoxyribonucleic acid. It's negatively charged. these proteins and proteins in general are positively charged, so they're basic. So you've got an acid and a base, and they have a high affinity to each other. So if you change the charge relationship between DNA and the protein, you can loosen the affinity or tighten the affinity. So just by ionic changes, you can change how easy it is to sort of get at this piece of DNA that's wrapped around this protein. And in addition, if you just, say, remove this histone octamer altogether, now all of a sudden you have a naked piece of DNA. So these histones provide for a way to sort of block that RNA polymerase to sort of get at the gene that may be wrapped around this protein. Please, yeah, please interrupt.
Okay,
the more
so
the questions, the
I'm
better.
on genetics right now, and I'm looking at karyotype. Okay, so, and my students are asking me different sizes. So basically, maybe one of the answers is that, so in chromosomes at 1 versus 21 or whatever it is, the larger versus the smaller, there would be more histones in that whole chromosome.
Potentially.
Is it because
So
of tightness of wrapping, or is it because of the amount of histones that are?
it's potentially both. So the human chromosomes are numbered 1 through 21 in order of decreasing length of the DNA sequence. So chromosome 1 is the biggest, and it's 50 megabases, million bases of DNA, and chromosome 21 is the shortest. But if you look at a karyotype, it is the composition of the overall length of DNA as well as its compaction. And depending on the packing density of these nucleosomes, you get a tighter or sort of fatter looking chromosome. Yes, go ahead.
One more question then. So I tell the kids that during interphase, as far as the cell cycle, interphase is when DNA can be read. It's like that naked
Right.
piece. And the only time it is condensed into chromosomes is
During mitosis.
Mitosis, that's the only time you can see that. So then there must be
Exactly.
there must be some signal there. During interphase, there's different things going on. So there must be a signal during one of the phases of interphase in order to get the.
Yes. So in late interphase, what happens is all of the histones across the entire genome lose a bunch of the chemical modifications that sort of decrease the charge difference. And instead, by losing those and by adding marks that sort of increases the charge difference, you get this tight compaction. And in addition, you get these yellow things that bind, which is another special histone called a linker. And what these linkers do is that they allow for this formation of what's called a solenoid structure, where these beads on a string wrap around each other. So instead of sort of a 10 nanometer fiber, which is this, so DNA is 2 nanometers, this is 10 nanometers, this is 30 nanometers, and then on top of these linker histones, you get a bunch of other proteins that act as scaffolds that you can form tighter and tighter protein-protein interactions. So it's really the histones that drive the physical compaction of DNA during the cell cycle. And so it's a very dynamic thing, especially in yeast, which is a eukaryote like our cells. The cell cycle only takes 90 minutes. So this entire process, every 90 minutes. All right, so we started touching on this, but really the stuff of epigenetics are what are called the readers, writers, and erasers, both of DNA and these histone proteins. So perhaps the most familiar epigenetic thing that you're familiar with is DNA methylation. So we're all familiar with A's, T's, C's, and G's, but actually there's a fifth base, which is a methylated cytosine. And this methylated cytosine is called the DNA methylation event, and it really only occurs on C's, and primarily C's that are flanked by G's. And the reason for that is that, remember, DNA is double-stranded, so that means on the other strand you have a C and a G. So it's sort of a symmetric pairing, and the mechanism's all worked out, but during DNA replication, when you make 2 copies, what happens is there's one protein that recognizes that this is actually a methylated C, and it tells another enzyme that's right on the other strand to methylate the new cytosine that just gets added during DNA replication. So this is a true epigenetic mark because it's propagated through every cell division event. All right, so what does DNA methylation actually do for the cell? It actually blocks proteins from recognizing sequences of DNA that it would otherwise be able to bind to, so these transcription factors. It also acts as sort of, this chemical acts as a binding surface for other proteins called methyl CG binding proteins that are really important for things that hopefully we'll get to. And as I mentioned, it's inherited with cell division. So this is sort of the canonical true epigenetic mark. And then we have the histones, and these can be covalently modified. As I mentioned, there are also variant histones that are just intrinsically have different structures. And so just you can only add so many modifications to a particular protein. So you can only, say, change it by 3 electrons worth of charge. Whereas if you just replace the protein entirely, you can get even more charge change. And then once you have this modification on a histone, there are also effector proteins that are called readers that can read out the state of a particular histone. And then in addition, there's a very new class of RNAs that don't code for proteins, but that are thought to act similar to transcription factors in that they'll recruit readers and other writers and erasers to particular regions in the genome, because the RNA itself can partially hybridize with a piece of DNA. So it's similar to, it gets transcribed off of a piece of DNA, and then it sort of partially hybridizes to that piece of DNA and then partially hybridizes to this protein. And so there's a variety of particular, so all these readers, the writers and erasers are enzymes, and they can, the particular chemical groups that they can add or remove are acetylases or methylases, blah, blah, blah. This is sort of something that you can read up on if it's relevant. And then for each of these 4 histones, typically the structure of each protein is there's sort of like a ball that's tightly packed, and then there's a, on the N-terminal end of the protein, there's this unstructured tail that literally extends out past the DNA coil. And this is the primary site of all of these chemical modifications. So the different flags represent different types of modification depending on the amino acid, and these can all be added and removed and read by reader proteins. So the general model, although it's not quite a correct term, is the histone code, that there are upwards of 100 different individual modifications that a given nucleosome could receive, and it's the combination of these particular modifications that provide an information readout for whether or not a gene should be expressed. And the idea is that DNA methylation and these histone modification enzymes and non-coding RNAs all cooperate to either block or allow these green proteins, which are transcription factors, to access the genes that might be encoded on this piece of DNA, or if you take off other modifications to sort of compact the DNA tighter together to prevent a protein from getting in. And that's the general idea, is sort of opening and closing of DNA to allow proteins to come in and transcribe a gene into RNA. Now the key is, how do you get a particular region of the genome to remember to stay closed? And that's really the memory aspect of it. And the memory aspect really comes in the concept of a feedback loop, right? So when you think about a feedback loop, the best example I can give is just the thermostat in your house, right? You say, I want it to be 72 in here. There's a sensor that senses the ambient temperature. If it's above or below that, it turns on some machinery that makes it hotter or cooler, and then it goes off again. So there are 2 primary types of feedback. One is positive feedback, and the idea is, let's say this is some gene that somehow perceives an environmental stimulus. That stimulus will turn on that gene. That gene will then turn on another gene and another gene. And what gene 3 does is it actually goes back to gene 1 and tells it to turn back on. So that once you turn this on, you can remove the initial stimulus and it'll keep cycling. right? It's sort of you get the ball rolling and then it'll keep rolling. Now, when we're talking about cell types, we really want what's a combination of this positive feedback with a negative feedback. So if this is a stem cell, what we want to happen is we want this to differentiate, but we want to prevent it from turning into something else. And so the idea is you have a stimulus, and initially this cell can go one of two ways, but eventually what will happen is this gene 3 here will actually repress one of the upstream genes that's required to turn a cell into, say, a blood cell. So it'll facilitate being a skin cell, but repress all the other cell fates. So it may take some time to digest, but something to think about. And the key is this happens both at just sort of the transcription factor level, but it also happens with these readers, writers, and erasers, right? So even just at a single gene in the genome, you can have a feedback loop with readers, writers, and erasers that maintains a particular set of chemical modifications. And then, so there are all these wonderful web tools that hopefully we'll get time to do a few demos, but there's links to them all. They work great. You can just search the human genome, the plant genome, all the genomes that have been sequenced. And there's efforts to annotate not just the primary. So this is the sort of highest level view of an entire chromosome. And each little section is marked off by how tightly compact it is in a karyotype. So these dark areas are the areas that would stain in a karyotype, okay? And so you have your primary DNA sequence if you actually zoom in enough, but you can also sort of annotate the entire genome with where each of these modifications are in a particular time, in a particular place, in a particular cell type, and whether or not that cell type is healthy or diseased. So there's tons of public data where you can just go online and look at some of this stuff and say, I'm interested in this gene that controls flowering time in plants, and you can just look at it and all the different conditions people have measured. So every single research study that has been federally funded by the US government, the data is publicly available and goes onto one of these websites. And these are just called genome browsers. And there's actually a major endeavor to sort of annotate all the variety of human cancer genomes as well. So you can go on and look at how sort of the epigenome is impacted by mutations at the DNA level. So with respect to interesting epigenetic phenomena in humans, one major one is X inactivation. So all females have 2 copies of the X chromosome, and males have 1 copy, right? So what happens in females is that in every cell of your body, one of them is completely shut down. It's completely packed up into what's called a bar body, right? And that compaction is organized by these histones and the particular modifications, right? So in early development, actually in like the 2 or 4 cell stage right after fertilization, one of the X chromosomes gets shut down, all right? And if it happens at the 4 cell stage and 2 of those cells choose, sister chromosome 1 to shut down and the other 2 cells choose sister chromosome 2 to shut down, then all of a sudden all the genes that may be heterozygous, so you have sort of different alleles, you're going to express different phenotypes in half your body, right? So this actually involves a small RNA that's called XIST, which activates a writer protein that adds a particular chemical modification to a histone that shuts down all the genes. So that's sort of the first step. And this XIST actually just lines the entire X chromosome, literally. So you can actually attach fluorescent molecules to this RNA and look at it in a cell, and it just lines the entire thing.
Wait, can I ask one question? So when you said like if there's 2, but if the signal comes at a later stage, were you saying that?
So it's random which copy of the X. So
It plays out differently like throughout your body?
Yes.
Like which X is?
And so the best example are calico cats, which are largely female. And what happens is the pigment gene, which naturally occurs in sort of these orange and black flavors, it's on the X chromosome. And so what happens is you get your zygote here, and then at the 4-cell stage, you'll randomly shut off one or the other copies of the X chromosome. And so now you'll have some cells that express the black pigment, some cells that express the orange pigment, and then that's what you see in the adult once all these cells grow up. And then a subset of these actually differentiate into sort of hair follicle cells. And
Does the inactivation usually happen at the 1 cell?
at the, I mean, 1, 2, 4, given our ability to look this early, it's usually early, but you know, it's like before 16, but
Right.
whether it's really supposed to be 2 or 4, it's unclear.
This is where our bio books are so behind, because I think we refer to this as just polygenetic traits.
Yes.
That's it.
Yeah,
That's
so
what we
this
teach
is-
the kids. There you go. That means there's just
Well,
a couple different genes.
hair color
that-
is polygenetic, but in the case of calico cats, this one gene is responsible for a large variety.
Polygenes, it's one gene. but
Yes.
there's 2 different
It's
types.
expressed
So it's
in a
very specific.
variety of
Yeah.
cells. So the other example is what's called imprinting, which is the fact that if you have the same piece of DNA, depending on whether you got it from your mother or you got it from your father, it's either going to be expressed or not. And so you can have examples of genes that are expressed on both copies of your chromosome, regardless of the parent of origin. You have copies where it's expressed just in the father, and just on the allele that you inherited from the father, and not the mother, and vice versa. And one of the most notable examples of this, so there's about 100 genes in the human genome that are well established to have a genomic imprinting effect. So parent of origin determines completely which allele gets expressed. Maybe more, maybe less, but one of the canonical examples that you can look up has to do with the production of an insulin-like growth factor. And it's supposed to be expressed it's supposed to be expressed in males and not females, and there's this interesting, complicated dynamic to make sure that that happens. And there are a variety of human diseases that result in imprinting defects where it's expressed in males when it's not supposed to be. For a lot of these things, Wikipedia is actually a really good place to start. And they have lots of links to primary literature. So I mentioned the agouti mouse, and I mentioned the honeybees. So you have your worker and your queen, and this is sort of a honeycomb, and the workers build out individual honeycombs where they want to turn the larva that's inside into a queen, and they fill it with this secreted product called royal jelly. And royal jelly not only contains a small molecule that blocks the ability to add DNA methylation, it also contains another small molecule that blocks the ability to remove certain chemicals from histones. And so disease often involves the dysregulation of normal gene expression, often via epigenetic processes. So you have aging, where cell types sort of forget what they are just because of natural and often stochastic changes in the expression of some of these readers, writers, and erasers. So just cell senescence itself typically results in the loss of histones completely. And so your genome tends to sort of open up as you get older, and it allows for the expression of more and more genes. And if you think of sort of a cartoon model where a stem cell would have every gene expressed, and all of your terminal cell types is having only specific genes expressed, you can imagine how aging is the #1 predictor of cancer, because your risk factor, exactly. In addition, a lot of cancers result from mutations in readers, writers, and erasers. So a lot of leukemias result in actually not mutating one of these writer genes, but fusing it with a gene that is a transcription factor that drives it to pro-proliferation genes. And other genes result in a variety of human syndromes. So a single mutation in this particular writer called CBP is responsible for causing Rubinson-Taybi syndrome, which results in a lot of memory defects, craniofacial developmental abnormalities. There are actually fly and mouse models of this, and I'm trying to develop the first sort of ant model for this as well. And another famous example of a human neurodegenerative syndrome is Rett syndrome, which is caused by mutations in the key protein that recognizes DNA methylation. And what's notable about Rett syndrome is that this protein is on the X chromosome, and so it predominantly affects women. And the idea is that it's also predominantly de novo mutation, so you don't inherit it from your parents. It happens as a de novo mutation early on in development, and because of this whole idea of X inactivation, you'll get a mixture of cells which either express the mutant copy or express the normal copy. So it's a highly complex trait.
Can you talk a little, you don't have to do it now, but for leukemias and methylation, some of the therapeutics.
I'll touch on it, yeah. And so this is actually a figure from a recent review paper on all the different readers, writers, and erasers that have been characterized as being mutated in human cancers. And so you can see that in the particular protein residues that they read, write, or erase on histones. There's a ton of them. They're characterized in terms of the type of modification on the histone and whether or not they result in sort of gains of functions or loss of function. And obviously, these would be wonderful drug targets, right? So not only is there a major research endeavor to annotate the human genome in sort of healthy and disease states with where all of these proteins are, especially if they're mutated, there's also a lot of sort of research into drug targets. And so this is just an example of a whole variety of drugs that are either in clinical trial or available for basic research that simply target one particular class of histone modifiers, and that is ones that add an acetyl group to lysines, right? And so there are, these are actually erasers. So HDAC stands for histone deacetylase. So they remove the acetyl group from histones. And there are 10 copies in the 10 different genes that encode an HDAC protein in the human genome. There's only 5 in ants, but they associate with a variety of complexes that are either specific to neurons or general to all cell types. And so there are a variety of drugs right now that are sensitive to a variety of these HDAC genes, but not a lot that specifically target one particular complex, which is really what you want, is you want to target the neuronal one for ones that are mutated in glioma or blastoma. So there's a huge effort to develop these specialized epigenetic drugs. So this is just sort of a generic slide on frontiers in epigenetic research. And I'm more involved in sort of basic research, but once sort of the basic mechanisms are understood and just sort of the prevalence of epigenetic phenomena in nature, then more in biomedical and translational research, there's, well, how do these things apply to disease? How do we identify therapeutic targets, the actual development of these drugs, and then testing them in patients? And all of this is happening at Penn and at other research institutions. But what I think is one of the best reasons to use model organisms, in particular social insects, and the best way to sort of tease apart nature versus nurture is through twin studies. Because, especially identical twins, have exactly the same genotype, not just in all the cells of their own body, but in 2 different people, right? And so in this case, you might have 2 identical twins that have different phenotypes, and it could be because they were exposed to different environments early on in life or at some point in life that can help you understand the role of epigenetics and sort of decoding that environmental difference. But in humans, at least, we can't really manipulate the system, as it were, to actually prove, was it epigenetics or was it something else? And this is really why model organisms are important, because we can go in and give them drugs, and it's okay if they get sick, and because we've made that judgment call. But there's an interesting article in National Geographic about twin studies in epigenetics and sort of a more scientific-oriented primary review. But this gets to ants, and a few more minutes. So ants have sort of, ant colonies or social insect colonies in general have been called the superorganism as a metaphor because a colony, in a colony, each ant is often nearly genetically identical, if not genetically identical. But they all take on specialized morphologies. So here you have a queen and the workers that tend to her and take care of the brood. They'll carry the brood into a special brood rearing chamber. Then you'll have sort of the next, these are the gametes, the next generation of sexuals that'll fly away from the colony. They're typically kept in their own little chamber, just like our gametes are separated from the environment as much as possible. And then you have the older ants that leave the colony and go get food and bring them back. So this ant colony is really a good metaphor for a complex multicellular organism like a human. And it's a giant twin study, right? So just in terms of ants, so an ant is a social insect. A social insect is a subset of insects generally belong to the family Hymenoptera, but termites are in a completely separate family. They're actually belong to the cockroach family. But ants, bees, and wasps, so every ant species is considered social. Only a very small subset of bees and wasps are social. But the idea is that in a social colony, you have a subset of individuals that are the reproductives, and the vast majority of individuals are considered workers that just sort of help the queen and take care of the brood, go out and get food, perform defense, keep a tidy nest. And ants are wonderful because if you use bees, bees actually have to go out and collect pollen and nectar, and so they need to go outside. Ants, we have, in our lab, we have 50,000 ants in a room that's a quarter of the size. And each of them is in a completely environmental controlled box. We can control their diet, we can control their genetics, and so it's just a wonderful system to use. And on top of that, all of the epigenetic proteins are conserved from insects all the way up through humans, but often in fewer copies. So as I mentioned, there's 10 of these HDAC genes in the human genome. There's only 5 in ants. So, my pitch is that social insects are the premier models for organism-level epigenetics, and they live in these cooperative multi-generational colonies. As far as insects go, they live a really long time. So this carpenter ant queen can live for 25 years, and yet her daughters, the workers, will live at most a year, and the queens have the potential to live 20 years, even though they're genetically highly related to each other. So colonies are primarily female. Every ant that you see walking around on the street is a female, and you really only see males during mating flights, and you'll see all these flying critters that don't fly as fast as flies. They're a little bit bigger, and they're really kind of dumb and just kind of fly slowly around. Those are the male ants. And the idea is that these daughters of a queen, so we use this carpenter ant species because this one queen is genetically diploid, and she'll mate with a single male once. Then she'll found a colony, and she'll collect tens of millions of sperm from that male and lay tens of millions of eggs over the course of her life. What's notable is that that male is not diploid, it's haploid, so that every single one of her daughters inherits the exact same copy of the paternal genome. And so they're actually more related than any two sisters and brothers that we would have. They're, on average, their relatedness is 0.75 instead of 0.5. And in fact, there are other ant species that people are developing as models where there's no sex period. In fact, all of the daughters are clones of one of the mother cells. So every single one of the daughters is genetically identical. So what we'd like to do is disentangle nature and nurture in a system where genetics is highly controlled. And the general rubric is this is understanding the epigenetic mechanisms underlying the different specialized behaviors of these ants is this field of behavioral epigenetics, and that's the topic of that review paper that everyone can peruse at their leisure. So this is a one-day-old queen-to-be that was birthed in our lab, and these are the workers that took her out of her silk pupal case. And what we do is every few days we'll paint all of the ants that are born that day a particular color, so we know their age within a day or two, and that we can also disentangle the effects of age on behavior. And so this is sort of a development series of these two insects, the minor and the major workers. And the queen lays an egg, and then it's actually the existing workers that differentially feed a particular larva so that she'll develop either into a major or a minor. Well, this is sort of, so we, at this point, there's no epigenetic difference at the larval stage, but there's a particular, so in these metamorphosizing insects, there's a variety of larval pulses that are called instars, and there's only a particular instar that's sensitive to nutritional input to make the major. And we can actually give, so what the nutrition does is it changes the regulation of certain hormones, and if we just apply those hormones ourselves in the lab, we can induce the production of these majors. And to give you a sense of how the body plan correlates with adult behavior, the minors do all of the foraging. So we can just track these 24 hours a day and look at who's coming out to feed on sugar water. They love sugar, right? It's all minors and no majors, right? So behavior correlates very strongly with the specific nutritional input that they received long ago prior to lots of cell division events that took place and an entire body plan restructuring that occurs during metamorphosis, which is when they hide in this silk cocoon. So, some of the stuff we do is we look at where all of these histone modifications are throughout the genome of the ant, and then we can ask, well, are there differences that allow us to distinguish one ant from the other, just using these histone modifications as labels? And this is just a plot that says yes, we can. And that applies not only to the whole body, but also specifically to the brain. And at this point, I can do brain dissections, and we're giving a variety of these drugs that are under clinical studies in people right now to ants without going through a lot of time and paperwork, and see whether we can regulate behavior. And in fact, some of these drugs are FDA approved for schizophrenic treatment and other behavioral disorders. So the idea is, can we use a combination of drugs that change the distribution or effectiveness of readers, writers, and erasers, and either try and make majors that way or try and change their behavior? And well, this is just sort of a fun little video. This is one of our behavior assays. You'll see this one ant that comes out and finds food. She's a particular kind of a worker called a scout, and she'll drink sugar, go back to her nest, tell her friends, and then all her friends are going to come out and drink now. So this is a particular behavioral class of the minor worker that's called the scout, and she's sort of the risk taker, and she's actually our model for drug addiction at the moment, because the risk takers actually have a lot more dopamine signaling, and dopamine is what is is the basis for cocaine addiction, and yada yada. But basically, if we give one of these HDAC inhibitor drugs to the ants, we can see that we can get them to forage a lot more than if they didn't have any drug at all. And in fact, we know it's working through this pathway, because if we give them an HDAC inhibitor drug, so something that blocks this protein, with something that blocks, so that will block the eraser. If we block the writer that adds the chemical that this thing blocks, we can restore the behavior back to baseline. Right? And then if we actually look at the genes that are expressed in the brains of the ants, we see that there's some subset of genes that actually get turned on when we open up the accessibility of those genes with blocking of this drug, and then those go back down again once we, so that's sort of the stuff that I'm working on right now. And I'm happy to end there and let you go on. I have just sort of 2 slides on what I'll call the Darwin versus Lamarck debate. And it really has to do with the modern interpretation are some of these epigenetic factors inherited from generation to generation in addition to our DNA, right? So if we can see that a Goody mouse that has a high folic acid diet, does the mother's diet affect her offspring? And there have been a lot of popularized articles in the last few years about phenomena like this. There are a few labs that are actually studying the potential for inherited epigenetic factors that give rise to human diseases in twin studies. And it's a very controversial topic. In the last 5 or so years, there have only been a few studies that have really rigorously looked at the molecular mechanisms. And some of the more notable and high-profile examples have been that if you feed male mice a high cholesterol diet, then 2 generations later, their sons will also have a high cholesterol diet. It's a male-only thing. It doesn't affect females. This idea of the flowering time in the spring for plants, that is also transgenerational, so that'll be sort of encoded in the pollen of a plant, and it'll be expressed as a phenotype the next generation. There's also this idea of an odor memory, and not only in mice, but I think there's been a study in flies now where if you expose them to certain fungicides, that in mice, not only it changes the offspring's mating preferences, but it can also lead to premature neurodegenerative disease. There's actually an example of a single-celled organism called Oxytricha. It's just found in sort of freshwater ponds, but its genome is kind of a mess. It doesn't have chromosomes. It encodes each gene on its own chromosome, but what it can do is it can take up RNA from the environment, and if that RNA is similar enough to a gene it already has, it will edit its genome based on the RNA from the environment and then pass that altered RNA from generation to generation. And one of the sort of my perspective questions is whether we can look at epigenetic inheritance of lifespan in ants, because in this carpenter ant we have the minor worker, the major worker, and the future queen, and we can keep them all separate in the lab. And one of the neat tricks about ants is that we can keep all of these ants virgin, and if we isolate them from their existing queen, they'll start to lay eggs that'll be unfertilized, and they'll be haploid, and they'll be male. So then we can look at the males that are generated from the major, the minor, and the queen, and males typically only live a couple weeks, so we can just see if they have different lifespans based on their parent of origin, which would clearly be an example of transgenerational epigenetics. So here are some more links about these examples, and people are interested, I'm happy to sort of show them some of the wonderful data gateways out there. But I'd say the most informative, so in terms of getting students excited, I think ants are a great entree into that. And here, this particular photographer, scientist, he actually has his own lab, Alex Wild. He's a great social insect photographer, and each photo comes with like blurbs about that particular species' lifestyle. But the NIH is the National Institute of Health is the main funder of taxpayer money, funneling it to basic research in the country. And everything that's funded by the NIH is publicly available. And there's a ton of free primary research literature for later high school students. There, you can just search information about any gene in the human genome, and then it also has links to if that gene is in any other genome of any other animal that's been sequenced. This website is also run by the NIH, is the online database for Mendelian inheritance in man, OMIM, and it has sort of descriptions, sort of cursory descriptions of all known human diseases and syndromes, but then links to primary literature. And this is one of these genome browsers that you can interactively just look at particular areas in the genome and all of the epigenome features that have been annotated. And then I'll try and, so this presentation on the first slide, in case nobody got it, I posted it on Dropbox so that you can look at it again. But I will post some of the videos I have here at the end separately. So this is another ant that we work with in the lab, and it's a predatory jumping ant. And so what happens is this ant who gets to become the queen is actually democratic. So if we remove an existing queen, the workers fight to become the new queen. And in a period of 10 days, they go through this fighting process, and whoever fights more actually kicks up their dopamine in the brain, and they sort of rewire their brain to thinking they're a queen, and that changes their hormone system. They turn on their ovaries, and within 10 days, they start laying eggs. And that process is reversible in as well. So we can turn an individual ant into a queen and then remove her, and she'll turn back into a worker. And what's neat about this is that the queen, the winner of this fight, gets the right to lay eggs, but she also gets to go from living 1 year to living 4 or 5 years. And she also gets to lose her eyesight, and her brain shrinks by about 25%. So we're looking into sort of what the basis of that.
In the first year or by the fourth?
We don't know yet. Well, it's not by the 4th year. It's definitely within 1 year. We don't know how quickly, though. Yeah. Let's see, this is a 1-day-old major ant. These carpenter ants have a very keen sense of smell, much better than us, and they can tell whether an ant came from their own nest or from a different nest, even though we keep the 2 nests right next to each other in the lab. And within a second, they'll be on them and police them, if not decapitate them. But what's neat about it is that we can take an ant, as long as she's within the first week of her life, from one nest and put her in a foster nest, and she'll be raised as if she belonged to that nest, and she'll actually become aggressive towards her true parents or true sisters. So there's some sort of odor learning, and then sort of the most socially sophisticated ant are thought to be these leaf cutters. They actually have 8 different morphological types that can be generated during early development, and each of them has sort of their own set of behaviors. So I was fortunate enough to go to Peru a year or 2 ago to study some of these. But they'll cut leaves. The bigger ants will carry the leaves down to the base of a tree and then drop them and then go back up the tree. And then there's other ants that'll take the leaves from the ground back to their nest. And then there's other ants that'll take care of the leaves in the fungal garden. But with some of these, on top of the leaf, you'll have very small ants that will swat away what are called decapitating flies that'll come in and try and knock the heads off of the ants that are carrying the leaves so that they can go and seed their progeny into the guts of the ant that they just decapitated. So the relationships of ants with nature is quite amazing. This is a photo of the leaf bed at the bottom of a tree. Here's an example of just the life cycle of an ant. So that egg gets laid, and after about a month, it goes through these larval stages and then enters into metamorphosis, and then anatomy of an ant. So if you guys have any other questions, I'm happy to hang out and talk, but certainly appreciate it.
The next one of these sounds cool.
I'm happy to go into a more specialized topic on any of these points, whenever.
So why not, since we are a little bit over time, why don't we thank Dan, anyone who has to leave can leave. Make a quick couple announcements about these things, and then if you have
Yeah, sure, I'm
minutes for
happy to
anyone
stay
who has
around.
questions. So that was a great talk.
Yeah.
Thanks, yeah.